officially have begun. Thank you very much, and we'll begin. Nevertheless, I want to finish up. There's very little about the nucleus, and really today is mostly about the cell cycle. I did erase that stuff. I'll go up and read. I'll go up and unerase it in just a sec. Housekeeping wise, it's unfortunate the class. This would have been better if we had it if they were not here on Wednesday for whatever reason. But so as soon as they get out, I'll be ready to start getting the exam started. We'll be pressed a little bit for time. I can probably, I'd like to give everybody an hour to do the exam. And if we need to do it, if you're not done by the time I have, my next class is in the same room. So I can either get them to come in 5, 10 minutes late. It's not a big deal. They don't have an exam, or I can carry it over into the lab. We'll figure it out. So I will try to get started, and you should have adequate time. So that's the note. It's just all days. Today was the day they weren't here, which is the first time I can remember, because they do tend to run long. So you guys who are just waiting out in the halls for them to sort of clear out. It's okay. We'll figure it out. We'll give you at it. I don't want you to feel an unnecessary time constraint. Would be the way to describe that. This is kind of where we left off. There's not a lot to say here. This is fairly straightforward. I'll put it off the blank. There we are. Make me look good here. Oh, sugar. It turned itself off. I'll say it. So I was going to say something inappropriate, which I almost did yesterday when we blew an 18-point lead, but all good things. This is a nucleus. So it gives you the basis. You're going to start to see it demonstrate itself over there. Largest organelle, we knew that. Genetic library, we knew that. Basically everything here. A couple of little quick notes. For our purposes, we're just going to look at one nucleus for the cells we refer to. It's a little bit easier. When we do bones, we're going to talk about multinucleate cells when they talk about this particular group of cells, which really are traditional bone cells. But and down the road in AP2, red blood cells aren't really cells either, because they shed their nucleus. They don't no longer need it. So for our purposes, it's one nucleus. But the big deal here is that there are three major components. The envelope itself. The envelope is interesting. It's a double membrane rather than a single membrane. Both sides are permeable. And I guess one way to look at the internal membrane gives it structure, and that external membrane is kind of like the connector to what's called the endomembrane system. Again, that's the system that starts with the nuclear membrane and continues on basically as a passageway for messenger RNA and for the materials and proteins that have to be protected within membrane as they sort of magically make their way through and eventually get utilized either in the cell or get excreted out of the cell. And so you can see there clearly the nuclear pores. That inner one has a structure, which we see in a lot of cells. They're sort of like the underpinnings that give the nucleus its shape, as it would for any other cell. That's there. You can see the nuclear pores, which again are very selective, but big factor is they're big enough for very large. RNA is a very large molecule, but it's linear. Where DNA, the fact that that is effectively a double molecule, that sort of spiral staircase orientation, so it's double-stranded, makes it too, let's think of it this way, it's wide or large, diameter-wise, to get through there. Everything else can get through. So whether it's things that are going in and things that are going out, it maintains the DNA in there. And that's a very unique feature of it. That's there. So it's an intricate part of the selective permeability. And then the nucleolus or nucleolus, doesn't matter how you pronounce it. Again, next test, standard question, what happens there if they make ribosomal RNA? No, frankly, no one is really 100% sure, but the consensus of opinion of authors and people who write textbooks and study this find that ribosomal RNA is being made there. And so it's pretty much SOP, standard operating procedure, to put that on a test question. Yeah, and I like to pick out ones that you might see again for licensure exams down the road. So it's trying to make one that's pretty, I've seen consistently. And then lastly, and perhaps the most importantly, is the chromatin. This is incorrect. This is the uncondensed version. Chromatin is a mixture of DNA and proteins, some other odds and ends, maybe some ribosomal RNA that's in there. What happens is it has a very indistinct, you can see it stains, but it's not very distinctive structures. When you see chromosomes, as we're going to discuss at the start of mitosis today, when you see the pairs of chromosomes, effectively they supercoil. Another word for that is to condense, and then they become visible. And here they're not visible because it's, if you think about what DNA is like, it's like a spring. It's like a coiled spring. When you stretch it out and don't allow it to spring back to it when it's in its original form, all spring and taut, it's super coiled. You can visualize it under a microscope. When it's stretched out and your proteins are in there keeping it stretched out, as you're going to learn, they're called histone, Histone proteins. They make it appear as chromatin. It's sort of like tiny threads, but without the distinctive nature that we've come along to associate with chromosomes, because most of you learned about meiosis and mitosis a little bit, and you saw the various stages of mitosis classically the way we teach it. We're reviewing that today as well. So that's really what chromatin is. You'll see it a little bit better. Nucleoplasm is just cytoplasm of another name. The inner layer is that lamina, gives it its shape. Nuclear pores or pore complexes. Regulatory, very selective in nature. There's nothing under the sun. Here you can see the pores. Here you can see that underlayer. This is sort of the look on the exterior of it. So there's not a whole lot else to say about it. Pretty straightforward. That's there. Here's the nucleoli. There can be more than one. Ribosomal RNA synthesis, a couple per cell. Chromatin. Surprisingly enough, that thread-like material is mostly protein, and those are the names of the proteins. We study them because when they keep the DNA stretched out, it can't replicate. It can't either undergo, we can't have cells replicating by way of mitosis, or it can't be expressing itself by making proteins. It's only when it supercoils. And that's very, very interesting. Okay, and so we're studying the histones because if we can keep it from replicating, it's one way we can try to control cells that are cancer cells. The big problem, and this is part of the thrust of today's lecture, is to understand mitosis is a normal process where we're designed to do what? Make identical copies, period. We're going to make all new cells that are absolutely the same as the existing cell. We'll start with one, we're going to make two. Those two are going to make four, et cetera, et cetera. And it's a continuous process where a lot of the cells are continually mitotic, just at different rates. The whole idea is they're going to be identical. If they're different, it's a problem. When do we want different cells with a similar process as meiosis? Sexual reproduction. That's all about diversity. We don't want the same thing. Okay, the whole idea of how species flourish is the basis of Darwin's theory, descent with modification. Okay, we have new generations. There's subtle changes. They're better suited to adapt to their environment. I don't do evolution, but you kind of get the idea. So today we're studying mitosis, all about these identical things. And one of the ways we see that's consistent with cancer cells is its mitosis gone out of control. So one way we can try to control it is keep those histone proteins in place. That's more of an aside. I'm trying to give you, again, some context with what we're doing. The chromosomes are the condensed version of the chromatin. That's better said. And the condensed state protects it during cellular division because it's a very busy process. And here they're showing you these are the proteins. So imagine this is a taut spring, and if you stretch out the little things, I didn't do it with the other classes. Think Slinky. Slinky? Yeah. It's a toy. Do you remember Slinkies? You all make them in plastic. We got ours in metal. And Mom and Dad were always yelling, look what you did to the Slinky, you stretched it out. Help me. I'm in touch with my inner Slinky. So my wife, she goes, "You're acting really childish." I go, I'm not. We do that routine almost on a daily basis. So you basically have, so what's happened here? Not only has it condensed, it's replicated. When you see this, again, this is for another lecture. Chromatids, we call them sister chromatids. They're identical pairs of this one chromosome has duplicated. So you actually have like 4 strands of DNA held together by that's called a centromere. That's where it's going to anchor itself on the spindle, and we're going to get to all of that. So that just gives you an idea of what it's meant to supercoil. Thankfully, we're done with that. Number 2. All this is about the cell cycle. I do not do a lot of this particular PowerPoint. The focus point is the cell cycle. There's a lot of stuff here on how DNA is replicated, how proteins are assembled. This is not, to me, it's beyond really the scope of the course. We had a long talk last year ago this time with nursing and health science about what we needed to include and what we did. And typically professors try to include stuff that's important to them, and they want to give you a lot of material. There's such a volume of material. We're doing this course that really probably could take, rather than 2 semesters, we could really do it justice in 3, but it's just not realistic to do it this way. So a lot of the stuff you really don't need to know. I don't feel, and that's sort of a consensus. Too much information. So understand this is all about the cell cycle. I don't know how you learned about it, but back in the day, the teacher went up when they would see all the slides with all the prophase and metaphase that we're going to get to, and they put a picture of a cell up there in a standard rectangle with a circle. That's the cell, and because we don't see any chromosomes, it's all spread out. That's interphase. The cell is resting, and whenever you start to see the start of mitosis, where the chromosomes would appear, and then they start to line up, and then they separate magically to have 2 cells. Now it's busy. Nothing could be further than the truth. The busiest part of the cell is interphase. It's assembling all the materials necessary to make an entirely new cell, all the components, all the DNA, all the membranes, all the proteins, all of that stuff. And at the same time, it's carrying on its regular job. If it's a digestive cell, it's bringing in stuff and processing. If it's a urinary cell, it's spitting out stuff we don't need, reabsorbing stuff we need. If it's in your defense system, it's making proteins to fight infections. You get the idea. The cell hasn't stopped being busy. It's just got more to do on top of that. So that's the mistaken notion. The mitotic phase, called M phase, is relatively short. Some cells that are very rapidly reproducing, it comes with an alarming amount of frequency. I mean, you shed all of your gut cells every 3 to 6 days. You're making billions and billions of cells every day. So imagine how rapidly some of those cells have to do things and how much energy is expended in doing so. And so that just gives you a context of it. So interphase, period from cell formation to cell division, when the cell carries out its routine activities and prepares for cell division. It's super busy. So it was such a mistaken notion until we began to understand how complex a process it was. Okay, and during interphase, nuclear material is uncondensed, clear in that chromatin state. And then there are the subphases, which include DNA replication. So these are the subphases. And then finally, they got it down right. This is called G1. It could be gap phase. Some authors call it growth 1. S for synthesis, making DNA. And G2 or gap 2 or growth 2, the final preparations for division. Yeah. The premise of today's presentation is about something called governance. Our bodies tend to do things very, very rapidly. We govern them and hold them in check. Our heart rate, without something called the autonomic nervous system, which we go into at the very end of this term, it's part of its job is to keep your heart rate under control. The healthier you are, it's called resting heart rate. Our resting heart rate without that nervous system working properly would be over 100. That's a pathway to danger. That's a pathway to heart attacks and strokes and high blood pressure and all sorts of damage. Normal resting heart rate for most of the people in this class, and for anybody that's healthy, 60 to 80. Okay? It's a very good, quick measure of checking your heart rate as it is now. I used to do it in all my labs. And I mean, it just gives you an idea of your overall health. Even if in athletes, it's even a little bit lower. Okay, because they're training and their hearts. It's all about the reserve you have to deal with stressful things. What holds that cell cycle from being so rapid, that heart from being so rapid, has to do with the autonomic nervous system. What holds the cell cycle, I misspoke for a minute, is something called checkpoints. And so we govern the checkpoint. The cell cycle that's going too fast, I don't know about you, but when I do stuff around the house too fast, I have a much greater tendency to do what? Make mistakes. Mistakes when you're making cells are usually bad. You make bad cells. And a lot of times, if there's mutations, read that as errors in DNA in bad cells, they can turn out to be cancers. How do we avoid those? We have all of these safeguarding mechanisms that our body will destroy a cell if it's obviously being made incorrectly. We have a capability of doing that. Like anything biologically, when you exhaust that capability, then some of the bad guys get through, and then they can cause a lot of trouble, mostly causing cancer. When does it get out of hand? When we do stuff to ourselves that we shouldn't. Idiots like me who smoke for 40 years. Thank you. Here's bladder cancer. Okay? People who drink excessively, killing their livers, another defensive mechanism. Don't exercise, don't eat properly, are under a lot of stress. Pick a zillion reasons that have an impact internally. That's what this is all about. You can't overtax your normal defensive mechanisms, the ones that keep things in check. If not, then that cell cycle begins to spin. And what happens to cancer cells, then the mechanism can't even control them. They replicate so rapidly. Moving on. So this is what it looks like. Now, it's not proportion. Every cell is different. They've drawing this because it's easy to read. Roughly 75-25. On average, the cells in your body spend anywhere from maybe as much as 25% being active in mitosis and about 10% for the ones that are able to replicate. And not all of them can replicate. We'll get to that in a second. So here are the phases. These are the very famous checkpoints. The first one, known as the restriction point, at near the end of G1, is surely the most important. That really identifies early on if the cell is either capable or needs to replicate, or if there's some inherent fault in that cell that it should not replicate. So to summarize that. The other mistake people make, mitotic phase does not mean cell division. Mitosis, the definition of mitosis, has nothing to do with cell division. It is nuclear division. Mitotic phase consists of mitosis, nuclear division, and cytokinesis, cellular division. The two are all part of, quote, M phase or mitotic phase. So be very aware of that situation. Again, fine points like this is in exams where you get fooled easily. I will say it 1,000 times. The most critical part of succeeding on exams is reading the question. When you're not sure of the answer, go back and reread the question, because that's how we're able to tell these things. Sure. Not one of my students. That's former students. So moving on. Here's what happens. So in G1 growth phase, it's just that. The cell is conducting normal business. It's replicating more membrane, more proteins, the components, all the A's, T's, G's, C's, deoxyriboses, blah, 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 blah. It's doing all this right. In the process, it's generating waste products. Those waste products effectively include a series of proteins that you will see shortly are called cyclins, Cyclins. Those bond with certain enzymes, and if you reach enough, the appropriate amount and appropriate type of those proteins, they bond with certain enzymes, and it causes this checkpoint to be released. Effectively, the cell cycle is like a big boulder you put on top of a hill. You've got something in front of it like a gate. And in fact, you have two gates, one for one hill and one for the next hill. If you remove that gate, it starts to go down and accelerate. So the first checkpoint is the one that has the gate with the strongest lock, whatever analogy works for you, and it doesn't prevent it from moving on into S phase and then even into the second growth phase. The single most important phase with regard to mutations occurring is S phase. That's where they occur because S phase is dedicated to assembling the duplicate strand of DNA. So before you start to see those chromosomes appearing, they are being made. They're just being held in an invisible way, microscopically speaking, by those histone proteins. So where do mutations occur? S phase, errors in DNA. Once that occurs, afterwards, now in growth 2 phase, it's really additional growth, making sure that they're chemically speaking, and that's really the major part of it. If mutations are present, they are recognized. There are mechanisms to correct them if they are correctable, as you will see. Something that we, sophisticated cells, advanced cells like ours, have that capability. Bacteria, a very minimal single-celled organism, has a very limited capability of correcting mistakes. We have something called DNA polymerase 3. You don't have to know it. You'll see the name without the Roman numeral 3. That's a very advanced system in eukaryotic cells like ours that is able to not only identify there's a mutation, but to correctly remove it. Remember, it's like I walked up and down in between the aisles, A's and T's, G's and C's. If you get one where there's incorrectly matched, a G and a G, which one is the right side? Is this the correct side? And that the mutation is the other way around. DNA polymerase 3 can identify not only the mutation, but which side needs to be removed. Which side, not that there is a mutation, but which side is the mutated, and replace that segment of DNA. It's something that a lot of different organisms cannot do. Bacteria mutate tremendously. The reason we have so many problems with bacteria today is because we don't have enough antibiotics to treat them. The bacteria are constantly mutating. Antibiotics can't keep up. The one you're going to hear the most about down the road when you get to micro is called MRSA. Staphylococcus aureus is the parent name of that. The mutation is called methicillin resistance. It means that it takes our ability to fight that is so limited that we are literally running out of antibiotics for that and other bacterial infections. Mutations in viruses. Inherently, RNA viruses and even DNA viruses to a lesser extent mutate all the time. RNA doesn't have any of those what we call proofreading mechanisms. So if you wonder why COVID keeps changing, it's going to. Why does the influenza keep changing? Because it has to. Those are viruses that always mutate. Once you understand the biology and the science behind it, it gives you a better idea of ways that you can combat it, and indeed, if you need to. I mean, frankly, this is opinion, not fact. Okay? There's a reason why, as we're finding out more and more, these vaccines are less and less effective against COVID and influenza. It mutates. That shot you got today might not cover the virus that's coming at you because they're mutating all the time. Again, whenever you're making a statement to somebody, if it's your opinion, state that it's your opinion. So if it's something that's factual, like the nucleolus is where ribosomal RNA is the agreed to be produced, then you don't have to necessarily qualify. So as we move on, that second checkpoint will come into play and not be released by those same cyclin chemicals or by a new set of cyclin chemicals until such time. And if it's not released, then effectively the cell will destroy itself. We have a very elegant mechanism of cells destroying itself we will not get to today because I wanted to conclude with that, as you will see. And then we get into all these phases, the phases of mitosis. So what happens when we get to DNA replication, and this is some of the stuff that I don't include with this. Effectively, you remember DNA, same thing you've seen a million times before, two identical strands, anti-parallel. It's called semi-conservative replication. It keeps very, very fresh. So effectively, it divides. We call that a replication fork. That forces the DNA to separate. The strands are identical, it's just directional. One's right side up, one's upside down, and each new cell is going to have one original strand and a duplicate. One original strand and a duplicate. That's what mitosis is about. It's called semi-conservative replication. We conserve half of the original DNA. Why? Because if you, in cloning, you'd have this, you'd make a photocopy. Then in cloning, imagine you took that photocopy and made a photocopy of that. And then the next photocopy. By the time you did about 20 photocopies of existing photocopies, it starts to fade. Try it yourself on a photocopy machine. It will start to fade, and in other words, the DNA degrades. By having semi-conservative replication, it limits the ability of degradation. So effectively, it keeps the DNA, relatively speaking, fresh. You don't have new and then new changing and new changing. You have a little bit of old and a little bit of new. So where it divides, it's called a replication fork. Where it replicates over those two areas that have separated is called a replication bubble. You don't have to know that. We're not doing cell and molecular biology, or the primers that are laid down. And this is where they talk about RNA polymerase and DNA polymerase. It would be unfair, not for any of the classes I teach. It would snow you under with some of this stuff. A bunch of names that you're never going to have to know. I promise you, I never had a patient in 25 years ask me about DNA polymerase or RNA polymerase. Never. Go figure. Here's what they said. Can you help me? Yeah, I can do that. Good. So here's the DNA polymerase. Basically, not only does all the things it's saying here, there's variations of it that can do the proofreading. It's really very useful in that regard. Okay? Interphase. So the end result of all of that, two identical daughter DNA molecules. One complete copy will give it to a new cell, while one is retained in the original cell. Semi-conservative replication. Now, you should know. Just demonstration for you. So what you're seeing, this would be the area where the fork is, where they split. The whole thing would be encased in a bubble. Thank goodness you don't have to know that. Not going to have to do that. Now we get to the important. So, continuously for growth and repair. When a cell no longer can grow, or what we call differentiate, when it's reached the end stage of its development, it goes into something called G sub 0. Try to get there. It might have been in a previous slide. So effectively in that G1, it will exit that G sub 1 if it has reached maximal development. This is good news for reasons and bad news for others. Nerve cells, heart muscle cells, skeletal muscle cells, by definition, have exited. They no longer will undergo mitosis. Why is that good news? If you're no longer in mitosis, it can't become cancerous. What's the bad news? The bad news is if it's damaged, it's irreplaceable. It will heal with scar tissue. If you damage heart muscle, it heals with scar tissue. We have a name for that, a heart attack. If you tear a hamstring, like you saw players on the football field, you were watching yesterday or Saturday or any time, and they would all of a sudden pull up and reach the back of your leg, most likely they tore some muscles in the hamstring. Those muscles are never coming back. They're going to heal with scar tissue. How extensive the tear was in the damage will determine if they can come back successfully and how long it will take. But those muscles are never going to the muscle may get better globally. That part of the muscle will never be repaired. If it happens in a nerve cell, we have a word for it, a stroke. Brain cells die. So understand, it's very clear that the cells have died. They will not be replaced. They'll be replaced by scar tissue. The extent of the damage and whether you're able to cope with the damage or whether you'll be altered by the damage is a whole second question. That's why these things become important. So they have to replicate occasionally. Again, we replace ourselves. We don't repair ourselves other than with scar tissue. Skeletal, cardiac, and nerve cells do not divide efficiently. They really don't divide at all. Damaged cells are replaced with scar tissue. Mitosis and cytokinesis, two distinct events. Same illustration you saw before. This is the whole idea. What's happening with these phases? Why do we have to know this stuff? I don't go into early or late. There are 4 events in prophase that occur. It effectively is observed microscopically. So things aren't magically disappeared. We just don't see them under the microscope. It's not like all of a sudden the nucleus emptied itself and it just magically came back together. It doesn't work. Okay. Prophase, 4 events. Metaphase, the easiest phase. M for middle. They line up down the middle of the cell. We'll see. Anaphase, the shortest phase. Part. Telophase, telophase, doesn't matter. Reversal, the first 4. What are the 4 steps? The acronym that I use, below the line, is PMAC. Prophase, metaphase, anaphase, and telophase. Chromosomes appear. They appear before they supercoil. Now they're visible. They were there the whole time, but now those histones are not attached firmly at cortisol. 2, the spindle. We have microtubules. We saw the animation. They assemble and disassemble. Here they assemble. The spindle appears. That's where the chromosomes are going to attach to eventually facilitate their division. 3, the aforementioned nucleolus had to do something else. It disappears. It's visually disappeared. What happens is that nugget of whatever is composed of proteins, RNA, whatever is in there, separates enough so it's visible. And lastly, certainly not least, nuclear membrane. Same thing, it's membrane. Membranes can open and close. So it can separate briefly and then form back. So the membrane is still there, it's just not attached. The fragments are not attached to one another. Those are the 4 events. I don't care about early or late, just the events. Chromatin condenses, forms the chromosomes. Visible is the key word. They're called sister chromatids. They're held together by the centromere. That area, the centrosome, and the area where you have the centrioles, they basically divide, go to one side of the cell or the other, start making the spindle out of, and again, the tubules are dissolved except when they assemble. That way we can see them. Subsequently, the nuclear envelope breaks up. Microtubules start to attach to those centromeres, blah, 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 blah. The nucleolus is gone. So, I mean, to me, this is a bad illustration because you can see that they try to represent threads, but they're usually not that visible. This is better. These are the sister chromatids. It's already paired up and replicated. The centromeres in the middle. You can see they call them asters. Those are the centrioles that have moved and are in the process of moving to here and to here. You always see basically the spindle starting to form this layer of sort of array is a better word, of microtubules on either side. And now the nuclear membrane gone. There's some fragments, nucleolus. There's probably fragments in there, just not showing it. That's it. Those are the 4 events. Metaphase, they line up in the middle. They call it the metaphase plate. Easiest one to identify microscopically, slam dunk. Next test. You can imagine that. Here's the question: prophase, metaphase, anaphase, telophase. And I'll have a list of what goes on. See? Just keep that in the back of your mind for next time. Anaphase, shortest. They pull apart. As they pull apart, typically toward the end of them pulling apart and separating, the cell is going to start to divide. Cytokinesis is going to start. And it's going to start to lead into the reversal of those 4 events. So here it is, anaphase. And telophase, when they stop moving, everything starts to reverse. Chromosomes, can't see them anymore. Nuclear membrane reforms, nucleolus reforms. In a sense, it's visible. And all those wonderful microtubules basically separate just like they did in the animation and dissolve within the solution. At that point, cytokinesis begins. It's highlighted by, and you'll see it in this or other illustrations, by this, what we visualize what's called a cleavage furrow. Effectively, you have this spherical wall. and actin filaments inside begin to sort of pull and tug on it. The best analogy for me is my failed efforts at trying to make a balloon puppet. Getting a balloon and you start to, I don't know, the end product of me making a balloon product is always the same. I'm terrible. My middle child can assemble a vehicle and disassemble it completely. I have no such capability. And so that cleavage furrow that you're seeing here, just think of it. You start to twist it, you begin to see that area where you can imagine two flexible structures dividing. Really, there's little actin filaments that facilitate that. I don't get a lot out of that particular illustration at all. So these are the signals. Go and stop, basically what they are. As long as that blocker is there, that restriction point, it's a stop sign. It doesn't become a go sign unless certain criteria are met. Why would we have to have that? We would have to have certain chemicals that will induce it to change. Those are those cyclins. They're also impacted by growth factors and other things that we get to mostly in endocrine. And you see a lot about that in AP2. And then most importantly, cells are interesting. They never get too big. If you looked at our cells and measured an individual cell and a similar cell in, say, a whale or in an elephant, they would be a little bit bigger, but not proportionally so. That has to do with surface to volume ratio. Cells never get giant because the cell has to be able to bring stuff in and push stuff out. It has to, in order to do metabolism, it has to bring nutrients in. It has to be able to expel waste products. In a room like this, okay, surface area is the area of the ceiling and the floor and the four walls. The volume is measuring length, width, and height. If we doubled the height, we would by a factor of 8 increase the volume. The surface area would not be increased that fast, even though we would have larger walls. Remember, the ceiling and the floor aren't changing at all. That's the idea of surface to volume ratio. If it gets too big a volume, if we have too big of a box, it can't get in from the ceiling or the floor or the walls. They're just too far away to service all the machinery inside the cell. That effectively is what surface to volume is. So when it reaches that area where it's getting too large for whatever reason, it's going to probably end up dividing. And what would stop it? Classically, and this is what cancer cells don't have, contact inhibition. If we go normal cells in culture in the lab on broad, it'll form a thin layer. Imagine having a beaker or a flask, and you could just see a thin layer of cells, of tissue forming. It wouldn't heap up, it wouldn't go down, it wouldn't have irregular humps and bumps. No, very level. Cancer cells just go unbelievably, ravenously and raggedly all over. Very disorganized. So contact inhibition, once the cell is striking other cells in their boundaries, chemical signals are basically saying, hey, enough growth. Imagine when you have a sore, it fills in the gap. When it grows over or insufficiently, there's a problem. So look at all the cuts and scrapes. They just stop magically when it's filled in. That's called contact inhibition. And you'll learn about that going forward. Those are the types of things you begin to see. You'll appreciate that. And I tell all the students, start looking if somebody in your family or you have cuts, scrapes, see how long it takes to heal. Look at the stages. Get an idea of what they're supposed to do, because then you'll be able to recognize when they're not doing what they're supposed to do. And that means a problem. So here are the cyclins and the cyclin-dependent kinases. That's the name of the enzymes. So the cyclins, and they're destroyed. Every time the cell goes through its cycle, they're destroyed. Okay? Every time. So it's a whole new ball game. After the cell is replicated once, we're going to start all over again. Each cell starts off with G1, S, and G2. So the checkpoints are critical. The G1 checkpoint is the one that is most restrictive. So effectively, all the building blocks have to be there, including the building blocks for DNA. And if that's met, then you'll have enough of these protein type, what I would call waste products, to create the ability to move that gateway and allow it to proceed. And then again, it may enter G sub 0 if it's reached its end product of being advanced, or what we call differentiation. If not, okay, a lot of times those cells, and there's a mechanism, again, we won't get to today, undergo programmed cell death. Basically, the cell is like, oh, this is a cell that we don't want to make. We're going to destroy it. Chemically, all of this is chemical signals. And so this is the part you don't have to know. This is all about what goes on, how DNA directs the order. It's very simple language. It only has 3-letter words. And you don't have to go into that. Basically, that's part of what becomes the genetic code. We talk in DNA, we talk about triplets. In RNA, we talk about in messenger RNA, we call them codons. In transfer RNA, we call them anticodons. We could get into the mechanics of this. It's just not worthwhile. So if you want to look through this, it's fairly straightforward. Okay, this is the neat thing about our messenger RNA is like this. It's called pre-messenger RNA in the nucleus. Yet, what leaves the nucleus is considerably shorter. We take out what we call insertion sequences. They're called introns. If mutations are on the introns, they don't affect us. That's the beauty of the way why our cells are less likely to develop mutations. We have elaborate proofreading mechanisms. We have what we call messenger RNA processing. We chop out pieces that's really DNA that just has very basic instructions. It doesn't have anything to do with making proteins, so there's less DNA to make wrong proteins. That's basically what mutations do. And lastly, we have the beauty of genetics, dominant and recessive. I mean, even if you have a recessive disease, only one side is expressed. So, I mean, or a dominant disease, it has to do with that. We don't always express exactly what's going on. So there's a lot of safeguards in the idea of having pairs of DNA, as opposed to something like bacteria, which just has one double strand of DNA. So I don't go through all of that. The roles are straightforward. Messenger basically has the message from DNA. It's called transcription. And everything else, the messenger RNA making proteins, is translation. You'll see that here. They summarize DNA information coded into messenger RNA, transcription, something to remember going forward. Translation is that same messenger RNA basically being assembled into polypeptide. Here's that processing that you really don't have to get into here, but here you can see how much it shortens just even in this little graphic example. And we're not going to go to the factors. We're not doing any of this. This is the intricacies of how we assemble the amino acids into the appropriate polypeptide or protein. Don't want to do it. And we see a bunch of stuff. One little side note, this is how we make a lot of antibodies in a short period of time. We have one strand of messenger RNA, it hooks up to multiple ribosomes. So you can make thousands, if not millions of molecules in minutes to fight an infection. Because there's so many spots where it's doing the same thing. And that is everything for us today. I'll be sending you a lecture on Friday that starts here, and then we're going to go basis in the bone. I'm going to start with this. Thank you. See everybody Wednesday.